Warmly welcome. Hello, everyone. I warmly welcome all of you on the IASNM MISSI webinar series. This is going to be a 45-50-minute talk followed by a brief Q&A sessions. And during the session, kindly mute your iPhones and other devices. And you can put your questions in the chat box if you want, or you can ask those questions live once the talk concludes. And now it's my absolute pleasure and honor to introduce Dr. David Andres, who is the Director of Research at Stanford Cyclotron and Radiochemistry Facility. He oversees operations at the recently commissioned state-of-the-art facility, supporting over 25 faculty with 30 IND drugs to advance clinical and academic research. Dr. Anders earned his Ph.D. degree in carbon-11 radiochemistry from Imperial College London with Professor Nicholas Long and Antony G. before completing a postdoctoral fellowship with late Dr. Sam Gambhir, focusing on aptamer binding agents. He joined CRF full-time in 2021 and became director in 2023, leading innovation in radiopharmaceutical production to meet Stanford's growing research and clinical needs. Without further ado, Dr. Anders.
Well, thank you very much for the kind introduction, Simon. It's great to talk to our colleagues in India and across the world. So thank you for the invite. I'm really excited to tell you what we get up to. Well, in the basement of one facility, and now I think the first ground floor facility of our new one. Let me share the screen. Yeah. Okay, so you can see my presentation. So I'm going to talk to you today about, I know that this field is very clued up on radiopharmaceuticals, but I'll tell you what we get up to basically with our cyclotron and how we move things from the beam to the bedside through radiochemistry. So this is an aerial photograph of the location of our newest facility at the 1701 Page Mill site, which was built with money donated by Sam Gambhir, which opened up officially in 2024. This was actually the old Theranos building, and so the cyclotron facility sits within that. And so today, I'll talk to you about cyclotrons, radiochemistry, particularly that around F18, C11, and then the ever-expanding field of radiometals and theranostics, and how these agents are moved into the clinic. So I guess Simon did a very kind introduction. So I grew up in a town called Stamford with an M, not Stanford. This isn't my house, but this is one of the stately homes of Burghley House. And then I did my undergraduate in Bristol. This is the Clifton Suspension Bridge, where I really discovered, I'd say, a love of chemistry and chemical transformations before doing my Ph.D. in Imperial College London, which was just across the water from the House of Parliament. So my research was basically doing electrochemical transformation of carbon dioxide to carbon monoxide before turning it into other drug molecules. This cyclotron was a little bit older than the one we're currently using at Stanford. And when the temperature was above a certain temperature in London, the cyclotron would have issues. And there was one particular day where we vented rather a lot of carbon-11 up the chimney, and you get a phone call from the House of Parliament asking if it was you were responsible. So that was an experience in itself. Now, I was fortunate enough in 2017 to join Sam's lab as a postdoc, where he worked on aptamers and multi-isotope PET. And then upon Sam's passing, did a brief interlude in Kathy Ferrara's lab, working on NK cell imaging and AAV tracking, before joining the facility full-time, where I oversaw the building of the new facility, installing a quality control system, a fully automated quality control system, and writing 6 new INDs. So the Cyclotron Radiochemistry Facility at Stanford aims to provide high-quality clinical and preclinical traces supporting our faculty and clinics. we do this under GMP to provide high quality tracers for our patients. At the same time, wanting to train the new generation of radiochemists so they can use these exciting techniques in their research. So at Stanford, these are the drugs that we currently have available for oncology. So gallium-RM2, SAMS tracers of Notin, FHBG, and DASA23, and FPPR-RGD2, and then other in red are the kind of tracers currently in development, as well as some antibody imaging and other metabolites. I guess neurology is probably our largest field of research. Someone recently said to me, is like half the population have a prostate, but all of them have a brain. So we do an awful lot of research in tau imaging, pain imaging with Sandeep Biswal, and DPA 713 with Michelle James, as well as O15 water studies and UCBJ. And this fluorodendromer, I think, was the newest tracer. In immunology, another tracer from Sam's lab was the OX40 tracer, along with Aragy. And this is actually the one I'm working on at the moment, which is, again, from Gayatri in Sam's lab, which was multitriose. And so, provide a good range of tracers for our faculty to use. So the Stanford CRF provides basically, we make $4.1 million, serving 27 PIs in 9 different departments. And at the moment, our FDG alone does 14,000 doses with 212 research doses. I mean, with that support, $84 million of faculty grants. So onto the kind of main body of the talk, where I'll talk about the kind of the production of radio tracers. And, one of the things I really love about my job is keeping this crazy train running. If you pinched me, I think, 10 years ago and said, you'd be producing drugs for the clinic in, an hour, and then I'd be like, you're crazy. But, this happens both at Stanford and in your facilities every day. And it starts off with the cyclotron, and then you produce the radio tracers, you do the quality control in under an hour, and then you release for injection. So everything starts with the cyclotron. The cyclotron was developed across the water from Stanford in Berkeley in the 1930s, more for, I guess, war than for peace, and the Manhattan Project. And these machines have really come on from being, the sizes of whole warehouses to these kind of, not quite benchtop, but the size of a small car. And they are, for the most part, incredibly reliable, accelerating charged particles, as does protons and deuterons. And they can be turned off at will, which makes them less scary than, I guess, a nuclear reactor. So we have at Stanford, we have a self-shielded one. The new one is a self-shielded one, and the older one from 2005 is located in a bunker. And they are fully computer controlled, and if it's working right, anyone can operate them. So the cyclotron into operation, you basically start off in the middle with your ion source, which is your hydrogen or deuteron gas. You ionize it, and it's accelerated by the d electrodes in a spiral path, where it's accelerated up to 10% of the speed of light by the time it reaches the outer range, where it's then extracted and flown into a target material of choice. So here we do your kind of nuclear reactions, and we represent our nuclear reaction with the target nucleus. So for carbon-11, you have nitrogen-14. You fire in your proton or a deuteron, and this is your recoiling particle, which is either a neutron or, in this case, an alpha particle, and this is your product. As you can see, we make a range of different isotopes on different targets, with half-lives varying from 2 minutes to days. This represents obviously new challenge as people go for longer and longer isotopes. I I was a radiochemist that started off with the kind of isotopes that if you had a problem, you just leave them a day and the problem was gone, to isotopes that one mistake and your lab is contaminated for months. So it's a certain change to our work and offers new challenges and opportunities. So our cyclotrons at Stanford produce fluorine, oxygen, nitrogen, carbon, with our newest solid target reaching kind of copper 64 and zirconium 89. I think one of the things I really love about my job is that it is the kind of modern alchemy, where basically the alchemists always wanted to turn lead into gold, where 1 gram of gold is about $85, 1 gram of enriched water is $35, and now recent discussions in research are basically trying to turn gold into mercury. So this is what a brand new cyclotron looks like, shiny and new. You have your vacuum chamber. This is a GE cyclotron. I know that the Siemens probably lies flat, but the principle is the same. If you have your vacuum, two-stage vacuum pumps, pumping this down to high vacuum, you have your copper D electrode, your ion source sits in the middle, and your carousels do your beam extraction into your target ports. One of they say that this part is the magnet. It's surrounded by a magnet, and this tunes this shape is basically dictated by relativity, so that as your particle accelerates out towards the speed of light, it gets heavier, and so the shape of this reflects that. And I think that's just fantastic that all of these different specialities in nuclear medicine come together to make these machines work. So these are kind of examples of gas targets, which are kind of long, thinner tubes that have your 60 milliliters of target material as gas phase, to your liquid targets, which just have 2.5 milliliters of kind of target material. And so that changes. I say that the cost of each target is to fill a gas target is $10, to fill a liquid target is $100, and to fill these newer solid targets, well, the cost can be $1,000 to $10,000, depending on your enriched material. And so this is our newest solid target installed on our second cyclotron at Stanford, and this will do gallium, copper, and zirconium, and scandium-44. And then you have, and it is all nuclear reactions are dictated by this binding curve, which basically states at different energies, you'll make your yield of different radiometals. And if you don't tune it correctly, you can get different impurities. So your beam energy must be highly tuned and homogeneous. Otherwise, you need really good purification chemistry. I'll get onto that in a minute. I was talking earlier with Jyotsna, and I think it is the thing that amazes me is the maintenance that goes into these machines. First of all, radiation breaks everything. And you need a dedicated team to keep these machines running 24 hours a day, 7 days a week. This is a very unhealthy looking vacuum pump, and my engineer on the phone to GE to try and fix it. And one of the other things I love about our field is how diverse it is, both in the specialities of the people needed, the backgrounds they come from, and in the size. You need your people that can crawl under things, people that can crawl on top of things, and to basically perform the maintenance on all these different parts. So on to the radiochemistry. So once we've made our isotopes, what are we going to do with them? So once a radioisotope has been produced, we try and incorporate it into our radio tracer. Everything is dictated by time. It must be done quickly due to the short half-life. So you don't want a synthesis that's more than 3 half-lives. So for O15 water, which has a 2-minute half-life, if your synthesis is over 6 minutes, that's too long. The other thing you're contending against is that we inject these things on the premise of low dose. So you need, if you're looking for high molar activity. So this is the ratio of radioactive to non-radioactive component in your radio tracer. So we're allowed to inject these things as diagnostics because we're going for tracer dose, and it's this value that is the assumption that's been made. Everything is performed in lead-shielded hot cells and can be manual, semi-automated, or fully automated. So the hot cells is basically your main workforce, and that these, because of the high radiation field, everything is done behind 7 centimeters of thick lead. It's always nice to see through, see your chemistry, and partly because people have used cameras, but cameras do not stand up well to radiation at all. And so your synthesis unit is located within the hot cell. So once you've set everything up, that's it. So you make sure you set it up and maintain it properly, because once you've delivered radioactivity to that hot cell, it's done for the day, or potentially the month with these longer isotopes. I will say I'll talk a little bit later about alpha, the theranostic isotopes, and how these things really change the way we think about, as a PET radiochemist, how we start to think about our workflow, because we have a different range of isotopes, from high gamma emitters to isotopes that have no gamma emission at all, but are incredibly toxic if you inhale them. And so the way our lab is designed is to basically, with the flow of materials, of going from dirty as far as the cyclotron is concerned, to clean, and then to shipping. And so this, so you have a flow of materials through your lab up until you're dispensing. And everything is obviously incredibly heavy. So most facilities are in the basement. I think our new cyclotron is one of the few that is not, but does have severe weight limits associated with it. And obviously, with the flow of your lab, you're trying to make things as streamlined as possible, because again, everything is a race against time. So in our lab, where we do a mixture of kind of research and clinical production, we have a range of synthesis units. So when we're developing chemistry, this is your kind of very manual, bespoke labeling, where you'll do kind of multiple reactions on small scales with not very much activity. And this is when you're kind of developing your method, changing this through to a kind of one patient dose, where it's kind of semi-automated. And then you have kind of more complex tracers that need kind of two-step chemistry, and again, used for kind of research doses, to your kind of full-scale manufacturing on a fully automated cassette that can produce basically 100 doses at a time. So I guess our chemistry has to reflect the variety of the agents that we label, from O15 water being one of the smallest, to an antibody of 150,000 Daltons. This range requires a huge range of different labeling methods to accommodate it. I always like to put the track in the trailer, because if you think about our label as the trailer and the agent as the tractor, if your trailer gets too big, you're going to have a huge knock-on effect on the tracer itself. I think one of the examples where this doesn't quite fit is FAPI, right? So FAPI is a small drug, but the label is almost as big as it, but it doesn't seem to affect the binding, which I find kind of fascinating. If you put kind of DOTA onto a small molecule in most other cases, you'd find that you really affect the binding property of it. So chemists need to come up with a range of different labels to label the range of different compounds that we make. So fluorine, natural products are rare, and this is great because, well, the drug companies kind of use fluorine to make them stable. FDG is used because of its lack of metabolism, and so you're kind of utilizing the strong CF bond to produce FDG, which does not break down in the cells to give you your fantastic images. It's also great because it has a low positron energy of 6.9 MeV volt, low positron energy, which means you have really good resolution images. It also has a half-life that basically means your expiry time can be up to 12 hours, which enables you to kind of ship your drugs around and gives you some time. They also tend to be pretty stable because nature is not very good at breaking down the CF bond. And so, and also because it's not naturally available, when I talk about carbon-11, one of the issues of carbon-11 is you have natural C11, which alters your molar activity. Whereas F18 drugs tend to be made with high molar activity because the sources of fluoride in nature are less and less. So I'm going to get onto my kind of basic chemistry now to tell you how we kind of make the drugs. And the first 5 of these points is basically describing how protons and electrons move. So you have electrophiles, which are compounds that like to accept a pair of electrons. Nucleophiles are compounds that like to donate their electrons. Acids are proton donors, and bases are proton acceptors. This pKa value is a key value for basically dictating how a compound likes to hold on to its protons or its electrons. This final term, the log P of how kind of fatty a molecule is also a key concept in making drugs. If your compound is too water-like, it just wants to be in the urine. It's like, don't need to interact with anything, just going to pee it out. And then if your drug molecule is too fatty, it basically sticks to the first bit of fat that it sees. And so you want to try and find a middle ground between these 2 points. And this is, I guess, the concept of spicy food. That if you have spicy food and you you drink milk rather than water to alleviate the heat, and that's why kind of milk works better because it interacts with the fattier spice molecule. Acid-base extraction is a key kind of purification technique. In the chemistry lab, you might have done this with a separating funnel. For radiation safety reasons, this is completely not acceptable. And so we've come up with these ways of basically doing this on these solid phase extraction cartridges. And I think this is kind of one of the subtle developments is that these solid phase resins can be built for anything. They're normally based off silica and then further modified to enable you to do solid acid base extraction or just separate things based off their log P. And so our other main method of purification is HPLC or high performance liquid chromatography, which again, I kind of like to compare to shopping on a high street. That if you go down a high street and there's lots of shops that you like, it will take you longer to move down the high street than if you're not interested in shopping at all. And this is how we kind of separate our molecules based off their affinity for the different stationary phases. So the first step in your kind of production, and I think one of the big steps in F18 chemistry was, I think when they started labeling things with F18, they looked at F2 gas and were like, that's going to be the best because everyone knows how reactive F2 is. But F2 has the problem that there's 2 of them. And when we do nuclear reactions, you're only going to make one part of it radioactive. So now most methods are based off the fluoride molecule. But the struggle with fluoride is making it more reactive because it basically wants to sit around water. And so we use this cryptofix compound here to basically separate it from the water molecules to make it really much more reactive. And so most of our chemistry is based off basically removing the water molecule. So the first 15 minutes of any chemistry is to effectively remove the water that it was made in and get it into the organic phase. And that's what these compounds do. Once you've got it into the organic phase, you basically mix it with your precursor. And your precursor as a radiochemist is the most important step in that you want to make our chemistry has to be fast and reliable. And your leaving group is the key component in enabling you to do this. So you basically have aromatic compounds, which tend to be much more stable. So the temperatures of these reactions tend to be 120 to 150 degrees Celsius. To your aliphatic, which tend to be more unstable, and these require quicker times. So you'll basically choose what's called a leaving group. And so this is what the fluoride is going to replace. And it will fall off if it can stabilize electronic negative electronic charge well. So, the halogens such as iodide, bromide are basically much better leaving groups than your fluoride. And even better still are your sulfonate leaving groups such as triflate. This is an example of our kind of, when you're trying to label biological kind of peptides, which aren't as thermally stable, you need to do what's called a two-step kind of chemistry synthesis. This was used for our alpha V beta 6 compound of notin. And so you do your kind of two-step labeling to make this kind of more reactive precursor that can go onto your peptide at a lower temperature. So I guess my first love in radiochemistry was carbon-11. The advantage of carbon is that it's present in all molecules. So you know that once you put a radioactive carbon onto your drug, you're not going to change its binding properties. This also, the disadvantage, as I was saying, is that now the disadvantage is that you have to be much faster. It has a higher positron energy, so it's less specific, and you also have you have to exude all natural carbon from your chemistry, otherwise you'll lose your molar activity, and you'll start kind of competing for your binding site with your non-radioactive drug. So C11 is made in the nitrogen gas target. So basically the first step is to take unreactive CO2 and pass it over hot metals with different, so either do a partial reduction to carbon monoxide or a full reduction to methane, where you then go on to hydrogen cyanide or methyl iodide. And methyl iodide is probably our most common chemical synthon, and the typical reaction time for this process is 5 minutes. Now I'll talk about, I guess the fastest growing part of our field is the use of radiometals. And the thing that really drives the radiometals is how tightly do they hold onto their chelating molecule. And this is dictated by this hard, soft, acid-base theory. So kind of when we talk about hard molecules, they're kind of small, high ionic charge, and they'll like to bind with other hard elements such as kind of oxygen. And then you have your kind of intermediate, they're not quite sure where they lie, and the soft are the kind of larger diffuse metals that interact with more like nitrogen kind of containing chelators. So, I mean, as far as I think there's a lot of work that can be done in the field. So we use these chelators for kind of larger biomolecules. As the labeling is often done at temperatures such as 37 degrees Celsius. The radiometals, like in main kind of stream use, are kind of gallium, where you have 68-minute half-life, copper, which is kind of intermediate at 12 hours, and zirconium, which is 78 hours. And so the aim is to match your half-life with the circulation half-life of your biomolecule. So for peptides, which circulate for like a month, you're looking at zirconium. For kind of smaller peptides, gallium 68, and for kind of intermediate proteins, copper 64. These chelators are kind of put on beforehand, again, low temperature conditions. In fact, with the biologics, I think this is actually one of from a regulatory point of view, from my diagram, I've shown just that we've put one dotted DFO, but the truth is that we have really no control over where we put these chelators. And so when you're making, if the FDA class your drug as a biologic, which is over 40,000 Daltons, it means it's really hard to translate these things past phase 1 trials, because they basically say, well, you can't fully characterize your antibody. So I think, site-specific labeling is kind of going to alleviate that. But right now, antibodies are definitely limited to kind of phase 1 studies. So once you've made your peptide, you'll put it into your mice, and you can see the effect that the different scaffolds have on the labeling. So this is the kind of clearance of alpha V beta 6, so 4,000 Daltons clears straight to the kidneys, whereas your larger molecular weight just circulates and ultimately ends up in the liver. So when you're thinking about theranostics, this is one of the really important considerations. So once you've made your drug and you've tested it in mice, it's time to get it into people. And at Stanford, one of the main routes that we use is the EIND. So this basically, because we acknowledge that our dose is a tracer dose, we kind of provide your first study is in small mammals, such as, I guess, mice or rats. And then you can basically go straight into humans. And that's your kind of second, your second species per se. And then, but the limitation is that, you are this, tracer dose, and the dosing can only last over 7 days. But it means that you can actually, under the EIND scheme, you can try different agents which are chemically similar. So this is the one that we did for DASA 23. And the first step is your toxicology. So the two parts of the IND that the CRF takes care of is the toxicology study and the CMC. So the first part of your section is the preclinical study that you need to kind of submit all of your data to the FDA on your animal studies and make sure that you do tell them about any adverse events. It's always better, in my experience, to be completely open with the FDA and when trying to move these things into the clinic. And then you'll go on to do your single dose toxicity study, where basically you inject a massive dose into your animals, and then after 14 days they are slaughtered, and you check the tissues for adverse events. So you'll kind of then submit these documents to the FDA along with the CMC package, which is the main part that the CRF takes on, and basically manufacturing things under GMP processes, which means that GMP, the FDA, is currently being updated for what are best practices as far as characterization and quality assurance. And it is basically, to make sure that our patients are safe. I mean, when we inject these drugs as diagnostics, we're allowed to basically just do a 1-hour QC rather than wait for the sterility, because we have to assure that our compounds are completely sterile. And so GMP practice means that we do our quality assurance means that we don't have to, the quality control is the same every time. I know that as a research chemist, that was the big change for me, was going like, well, if it shows up in the quality control, it's okay. As far as the FDA are concerned, that's already too late. So you just, everything, all the steps you put into the process are to make sure that it never fails the QC. And so that basically means that your people are fully trained and competent, and your processes and your SOPs are robust, and that they are ultimately followed, and that the products meet the quality. You write out the standards, and your products must meet those standards every time. If they don't, you have to do a full investigation. Your premises must be clean and maintained. So you're not only maintaining your cyclotron, but you're maintaining every machine, the HVAC system, all of it has to, work as stated. And to do this, you create, standard operating procedures, implement these. And the big one is then, whichever it's easy to make an SOP, I'd say it's harder to follow them. So you make sure that everyone can follow them, and you document when there's been deviations from procedures and the change control that you've put in place. So you make sure that these SOPs work time and time again to make the chemistry repeatable. And part of that is maintaining your systems and your facilities, and make sure your workforce is fully trained. And everything must be absolutely sterile. So this brings us to kind of theranostics, which what is, I think, a really exciting time in our field. But it certainly brings new challenges with regards to radiation safety, and now that we've moved from kind of diagnostic to therapy, all of the things that we've put in place as far as quality go up a notch. So new isotopes mean new containment challenges. I mean, right now the discussion is around actinium-225, lead-212, and astatine-211. Just within these three isotopes, you have three completely different kind of operating purviews. Like lead has high gamma emission. Astatine, so lead needs a lot of lead. Astatine is volatile and radiotoxic, and actinium is just incredibly toxic. And so your facility design, and the half-lives change. So your facility design, you have to, I think, completely change the way we've thought about production to maintain operability against things like contamination, etc. And waste is the kind of the thing that no one really likes to talk about. But with these agents, you often have kind of long-lived contaminants which have to be disposed of, and the waste very quickly builds up, particularly if you have a spill. Well, that brings me basically to the end. So I hope you've learned about cyclotron operations and how the radiochemistry of lighter elements such as F18 and C11 can produce radio tracers. And radiometals offer a new frontier in both imaging and therapy. And that your translation ultimately relies on reliable chemistry and good preclinical data. Well, yeah, thanks so much for listening and having me talk. If you have any questions, I'm happy to take them now or over email later. Thanks a lot.
Thank you, Dr. Anders. Dr. Vedmulsa, I'm one of the physicians at Emory, and Saima had to step up a bit for some engagements. So I'm taking over for the Q&A session. Thank you very much for the very comprehensive talk. It was a very good experience going through the nuts and bolts of radiochemistry, especially useful from the lens of a physician. We have a question in the chat from Manish Dikshit. I will read that out for you. He's asking, what are factors affecting choline-11 production and conversion to 11-choline CH3 via, I'm assuming this is lithium AIH4 and HI methodology. We are sometimes facing consistent yield issues at GPGI, Lucknow, India.
Yeah, that's a great question. I mean, yeah, carbon-11 is, I think, a specialty all in itself. Yeah, so, I mean, I think the liquid method, the limitation is this, ultimately it is specific activity is probably the issue a lot of you're running into. So I guess, I mean, so the liquid method means you keep things basically around room temperature or colder, right? But you kind of face that lithium aluminium hydride is incredibly reactive. And that means it can probably decompose quite quickly, I imagine. And you probably get it, unless you have to be very careful how you store it, in that you must really keep it away from the air. Like any carbon dioxide ingress into any of those systems causes huge problems. And I think the liquid method probably suffers from that more than the dry method. Although the dry method does suffer, it's just easier for us to bake and condition them. So when we use the high temperature method, so we typically use the high temperature gas phase methods, because first of all, the high pressure target that we operate, and you can kind of bake it and condition it easier than the lithium aluminium hydride. I think is a little harder to handle, to be honest. Does that answer your question?
Is Anil from India. David, may I have a question?
Yeah.
Yeah. So my question is that in the solid target, the production of any isotope that we are looking for the metal ions, mostly for the
Yeah.
copper 64 and 61.
Yeah.
As well as the comparison with the liquid target for the gallium 68. What is your opinion in this, and which modality is coming forward? This is the one related to the cyclotron. Second question is that when we are doing the radio labeling for macromolecules like antibodies and peptides, what I see many people, they do labeling at the same time when peptide and antibody also behaves like a chelating system. So there is no pre-labeling. Directly they conjugate, either they label and they do the labeling after conjugation. So what is your opinion that it should be done first, labeling, purification, and then conjugation?
Good question. I mean, I think it's always probably better to do the conjugation and then the labeling, because the FDA will always ask you for the kind of characterization of your precursor. So if you don't, I think in those kind of methods that you're seeing of the kind of clinical, they're kind of not doing that. And I think I think the questions on that will increase from the FDA and the scrutiny of those kind of methods.
Okay. So my only the worry is that when we do the conjugation, the labeling, we do not know the nature of the complex. Is cationic, anionic, either it is binding somewhere else. Like we see a lot of the PSMA molecules. They are pouring the metal ions inside. They don't know it goes to DOTA. Either it goes the behavior that you have already, peptides behaves in the same manner as DOTA. So this is what I wanted to know. What is your opinion in this for the conjugation before labeling, either after doing the conjugation after labeling?
Oh, I mean, so are you saying, I mean, is the issue that you're having is that you're getting radio metal, like other metals stuck in your chelator?
Is not. Yeah, means for example, if I use copper 64, 61, actinium, either any metal ion, lutetium, for example. So if I'm loading with already conjugated DOTA with peptide, either PSMA like molecule, where gallium, either lutetium goes, either in DOTA, either in peptide.
Oh, well, you mean without the DOTA, does it bind to the peptide?
Yes.
Yeah, no. So I guess the way you test for that is when you do your labeling, you're kind of mixed with kind of like an EDTA to find how specific your binding is. If you're not bound to the chelator, the EDTA challenge will
But EDTA is not
reveal
specific. EDTA is not specific because it's a acyclic system. Is good for trapping the iron, either something. But when we are competing with the DOTA and a cyclic peptide, then it will be like a competition between the DOTA and cyclic peptide.
That's it. Yeah. Yeah. No,
We'll
it's
chat
an
more,
interesting.
David. Just I wanted to know that, man, what is your opinion? Because I like your beautiful journey starting from cyclotron to really let everybody knows that what goes from beam to bedside.
Yeah. Yeah.
Beautiful.
No, it's an interesting question as far as the cyclic peptides. I still don't know the answer, but I'm excited to see any data that shows that.
Yeah. Yeah. Thank you very much, David. Yeah.
Yeah, Harshad.
Hey, hi. Hi, David. Thank you so much for the enlightening talk, Harshad from BAMF Health in Grand Rapids. This was really enlightening for majority of the audience is also physicians. So I would like to ask a more clinical question, not a very technical one. Would love to know your thoughts about choosing the right theranostic pair. So we know that gallium 68, lutetium 177, it was similar radiochemistry, go kind of hand in hand. Copper is, of course, in my mind, is the best theranostic pair for 64 and 67. So how do you choose, and do you have challenges in once you have proven the clinical use using, let's say, a gallium-68 compound? Is it almost a given that it could be radio labeled as well with lutetium-177 and or actinium-225, or just would like to know your thoughts on how do you select based on what peptide is or antibody is the right radioisotope and then the theranostic pair. Thank you.
That is a good question. I think that's a question that I think the field is still definitely trying to answer. I mean, as a chemist, I believe there's a huge difference between, let's say, gallium-68 and lutetium-177, just in the chelation alone affecting the binding of your compound, particularly for peptides. Antibodies are probably less influenced by that. But I think, first of all, antibodies don't particularly work well as a therapy at the moment, unless you look at the kind of click chemistry methodologies. Yeah, I think gallium-68 is used because it was like the first available. I don't think it's personally the best isotope, but it is one that we can make in kind of high volumes at the moment. And I think obviously the success of the theranostics field was driven by the gallium to lutetium jump. Are there better ones out there? Time will tell. I personally like scandium 44 and 47 if the production can be figured out. But as far as and I think also the different isotopes, I don't think one isotope fits all for the therapy part. You're going to need a range of alpha, beta, OJ emitters, different energies. I'm sure will have their own effects if it's probed. And as far as stability is concerned, I don't think I'll ever be fully convinced that actinium is stable in whatever chelator it's put in, just because of the nature of that decay, as far as alpha decay is concerned. So let's see. But yeah, I think there's so much work to do in this field, is one of the things that makes it so exciting as far as which pair is the best. Yeah, time will tell. And I think there's plenty of space to research that.
Hey,
Harshad, this is Amol. I think to add to that, it's not just the ideal pair, but it's also what it's eventually going to be able to commercialize. Because the ideal pair may be in the setting of a research lab, like Stanford has a lot of ideal pairs, probably.
Mm-hmm.
But once it gets FDA approved, you have the US is a big country, and then you also have the global perspective. And so, what would be most easily feasible to make it available to the masses would also determine the choice of the agent, actually. I think that plays a bigger role in what eventually becomes available more universally than what's ideal in the lab.
Yeah, I totally agree.
Yeah,
sorry.
Yeah, so I completely agree with you. I mean, I think, with copper-64 becoming incredibly successful, you're seeing, isn't it, stockpiles of the enriched material, can supply keep up with demand? I mean, and if not, then the price will increase dramatically. So, yeah, no, I think it's an interesting time just to, try these things. And if one thing takes off, particularly in the metals, and this happened in kind of, not just radio metals, but in, isn't it, catalysis? People moved away from rhodium to iridium because rhodium was scarce and now iridium is scarce. So, if something becomes successful, then the price changes and everything changes again. So, yeah, let's see.
Yeah, just want to add to it. I mean, completely agree that what is feasible and the supply chain availability, etc. And then looking forward to what would be the most feasible for commercialization. But my question was mainly like when you're choosing, of course, scandium and copper both being the same elements. So you're kind of the radio labeling is almost a given that if it works with scandium 44, it's going to work with scandium 47, and the same for the copper theranostic pair. But for most of these, because gallium-68, as you said, David, is kind of readily available, and so most kind of preclinical scientists also use gallium-68. And so then translating into, let's say, lutetium-177, that is somewhat similar, but with actinium-225, then there's a possibility of also the radiolysis and all this kind of stability of those compounds. So it's a very interesting kind of conversation.
Yeah, absolutely.
David, I had a couple of questions more from a clinical perspective as well. You talked about a lot of good things that we usually don't consider as physicians. One of the things that routinely happens is the patient shows up late or something happens and the patient does not get injected in time and the dose is slightly lower. If it's lower than what your RAM license allows, plus or minus 10% or 20%, whatever your RAM license is, you have to sign off. And we usually just we tell the techs to, see what kind of counts you're getting. And if you're getting, image a little bit longer per bed position so that you get adequate counts and so on and so forth. But is there a good, we never consider molar activity in that decision making process. And is there a good molar activity, and does it affect biodistribution? We don't know much about molar activity. We just assume that it is okay, and it won't affect biodistribution. And whether it's different for, say, F18 based compounds versus gallium-68 or other compounds, or whether it's different for diagnostics versus therapeutics, because we've ended up treating a Pluvicto dose the next day actually because the patient did not show up and so on and so forth. So how should we consider molar activity in such considerations?
That's a great question. I think this is like the radiochemists and the clinicians.
Yes.
Yeah, people I've spoken to, I remember, I mean, it's got to the point where even, some of these companies such as Lantheus don't even want to consider molar activity because it increases the cost of your operation. But for me, molar activity whenever you're talking about a receptor binding moiety, now depending on how much of your receptor is expressed is whether the molar activity is an issue or not. So if your thing is highly expressed, then you're not going to fully bind all of it. But in some cases, you kind of think you might. So, yeah, at the moment, I know the clinic doesn't worry about it. And I know when the theranostic doses of lutetium certainly don't worry about it. But I think we're not getting the most out of the therapy agents if we don't consider it. The reason we don't consider it is because of probably cost, right? Lutathera and Pluvicto are administered as basically single, unit doses because it's cheaper to produce that way. But are we getting the most out of these personalized medicines if we're doing that? I don't think so, but it does make workflows easier. Is it? I mean,
I think it's just a convenience, actually.
Yeah, I think it's more and I think we could get more out of it. It's just the workflows that we work within don't allow it at the moment.
Yeah, for a long time we were just like we just basically continued what we used to do with FDG. And for FDG it doesn't matter much, but I think for receptor-based tracers, especially like FES, I think it may matter more because already the intensity is on the lower side. And it can. And yeah, I had not considered the fact about the expression and lack of saturation. I thought we had a very good luxury that we have a significantly more injection than the express density of the receptors express. Is that not the case?
I mean, I guess the amount of actual kind of cold compound is never like much. It's like micrograms, right? It is tracer amounts. So that's the pretense you're working under. So you're kind of basically saying to the well, we never saturate binding. I know for for the antibody studies, you actually inject cold dose to basically saturate some of the receptors anyway, right? I mean, that's yeah. So are you reaching saturation? I don't think so. But for certain I mean, this is the kind of immune imaging. I think immune imaging doesn't even need saturation of a receptor to have an effect. And I think this is the example of our OX40 agent, for example. But yeah, I
Some food for thought,
don't know
actually.
enough, I think.
But one more factor to consider would be also the off-target binding, right? Because for diagnostic agents, if we are not saturating tumor, yes, fair, but we are also not saturating the physiological distribution. And for any extra molar concentration of radioactivity, we are going to deliver more radiation to the off-label targeting. And the same goes in a worse way for theranostics agents, that we end up radiating more of normal organs. So are there any techniques to reduce the off-target binding? Like PSMA not going to salivaries. I mean, if we can do that, actinium becomes much more important tracer, like immediately, not tracer, I'm sorry, the theranostic agent.
Yeah, I think those kind of modifications are huge for reducing, and basically makes the difference of which agents are ultimately becoming the mainstream. Yeah, how those modifications or different isoforms is, yeah, breathtaking, staggering to me. But
Yeah,
yeah,
actually, there was a
I love that about
company.
biology, right? That small changes can make huge differences in the response.
Yeah, I think there was a company. I think they were doing a trillium pre-treatment to decrease salivary gland uptake with PSMA. I don't know where it went, but tagging on to the same thing about a QA/QC, especially in setups where the cyclotron is in-house and you're doing in human use. Lots of times what happens, and I think you alluded to it, is sometimes the QA/QC process fails, but you come to know about it after the dose has been either released or sometimes injected. And in those cases, what should be the clinician be aware of? I mean, it's happened to me. I used to be in Louisiana. We had a cyclotron right next to my office with a GMP compliant facility. We used to make our own doses. And, I was also in charge of radiation safety there. So I've dealt with some of these hassles. But in general, we don't teach this. We're not much aware of this. So, and I would like our residents to know a little bit more as more and more are going to the community and are getting into these roles. So I just wanted to pick your thoughts on that thing as well.
I mean, within the US, and I know every regulatory authority probably has a similar, that within your procedures, you'd have what's called a kind of product recall. And I know the FDA certainly, they just want to make sure that these procedures are in place, that if something's been injected that shouldn't, I mean, ours kind of, basically it's put onto the physician ultimately. And but there must be some notification of the FDA as well, because it suggests that procedures, there has been some sort of procedural deviation, and that should be recorded if that has happened. And it does happen, but it just needs to be recorded, I think. And it is kind of, with as far as like FDA and any regulatory authority compliance, it is just, having the procedures in place, but the reporting in place that is put in place. And I think what's hard is in these kind of fields, it's kind of taking responsibility for a mistake is a very difficult human thing. And these procedures are in place to kind of document the kind of learning of kind of, okay, that shouldn't have happened. What are we doing to make sure it doesn't happen again? And that just has to be documented fully. And so you may have like recall procedures, and those procedures should be followed. If they're not followed, then you record it as a deviation and ultimately update your procedures to make sure it doesn't happen again. as far as the, I mean, depending on whatever the QC fail was, will have different kind of implications as far as safety. But on the whole, I mean, for us, what we had, a batch of FDG that failed a pH, but the pH test is an easy test. I think what's hard in anyone that follows procedures a long time, what happens is people become accustomed to a way of thinking, right? And it's hard to keep the workforce, and that's basically where you need the second checker, right? It's somebody always has to check somebody's work. And again, that makes people feel uncomfortable, I think, but I think it is completely necessary. And I know within like the big difference between when you're making a diagnostic and when you're making a therapy is the staffing amount that you have your checks in place. Because there's nothing worse than if you're making a drug and you're checking your own work, when you've put 6 hours of effort into something and it fails, being able to make that call, your judgment can be clouded. And so the systems must be in place to prevent that happening.
Yeah, especially at 4 o'clock in the morning.
Yeah, there's nothing worse than ringing up a physician and saying, I'm sorry, your 100 doses are not available. That's a hard conversation to have, but it's sometimes necessary.
I agree. Actually, I think there are a couple of comments, and so you might want to
Oh, yeah.
take one.
We have just, I think, another question from, I think, probably a physician from Tanzania. They're asking, what are the ways to improve the yield of F18 FDG and gallium-68 radiopharmaceutical? They are employing these two pharmaceuticals currently.
Oh, so I guess the question is, what is your yield of FDG? I mean, typically our yields for FDG is 60%. How do you improve that? Often it's it's incredibly specific. It depends, first of all, how you're making it, like what module are you making it on? A lot of it will be like precursor amounts. Just making sure, I mean, if you're making it on a cassette, the things that you want to check is obviously the cyclotron. I mean, we'll do multiple kind of what we call target rinses before we begin. Rinsing those lines out, making sure the delivery is effective of the cyclotron. It can be a whole number of things. And then for radio metals, it's just ensuring that there's no other metal contaminants in your process. And I always encourage my chemists, like with these automated modules, you always want to you've really got to watch them closely. Like at any point, you can have a very weird, like from anything, when you're piercing a scepter, for example, that a reagent isn't leaking out on you, or that bit of rubber hasn't blocked the needle. And that can happen. And that's where the GMP process of just really tracking your batches of things you're using really becomes important. And I would encourage every chemist to do, trend analysis. And I think that's really helpful. Just every production tracking your yield, which reagents went in. And I think this is actually one of the great uses of AI, to be honest. Like it can do this. It can put these kind of techniques into, everybody's hands. And all of it. Yeah, it's
Okay.
Yeah.
I think that's all the questions that I have in the chat. And I think we are also at the end of our time for today's IASNM Flashpoint webinar. Thank you, Dr. Anders, again a lot for participating and going through all of this with us. This was a very enlightening session. And I hope that we cross paths, all of us, at some point with you in person. And thank you for the audience for your insightful questions and participating in today's webinar. And all of you have a great weekend ahead.
Yeah, thank you very much for inviting me. It was great to talk to all of you.
Thanks, David. Thanks, Akshay, for taking over for Simon.
Thank you.
Thank you.
Thank you.
Thank you all for joining and for an amazing talk.
Thank you.